Genome-Wide Purification of Extrachromosomal Circular DNA from Eukaryotic Cells.
Hi, I'm Rasmus. Here I will present the overall outline for the method for circular DNA purification. In the first part, yeast cells are cultured and harvested. Then, circular DNA is purified and amplified in 3 steps. First, by column separation, where DNA circles are purified and enriched. Then, by treatment of DNA with X nuclease to digest all remaining linear DNA. And finally, by amplification of DNA circles using the 529 polymerase. In the 5th part of this protocol, DNA circles are sequenced and reads are mapped to the Saccharomyces cerevisiae reference genome. In the end of this video, we will show you some representative results. We will discuss the perspectives for future studies and the advantages for using this method.
Part 1. Cell Harvesting. The starter culture is grown at 30 degrees with agitation to maximum density, which is approximately 200 million cells per milliliter, and is reached after 24 to 48 hours. The outgrown culture is transferred to a falcon tube and pelleted by centrifugation for 3 minutes. The supernatant is removed. The large pellet is vortexed in TE buffer and repelleted by centrifugation. Part 2. Column Purification. In this step, we use a commercially available column purification kit that yields